How do you make a fossil? As you know, a fossil is any remains or traces of an ancient organism preserved in the earth. Paleontologists usually draw the line at 10,000 years. The remains or traces of an organism must be older than 10,000 years old in order to be considered a fossil. It must come from a time before the written history of human civilization. The study of fossils and what they tell us about ancient prehistoric life is called paleontology. Geologists distinguish between three types of fossils: body fossils, trace fossils, and chemical fossils, or biomarkers. Body fossils are, well, fossils of bodily remains. When you think about fossils, you generally picture body fossils. They are the first to come to mind. Trace fossils are fossils of behavior. Some of the most common trace fossils are burrows, trails, tracks, and footprints left by ancient organisms as they moved over soft sediment. Sometimes these trace fossils of tracks and footprints occur by themselves. Other times, they occur with body fossils of the organisms that left them. This horseshoe crab died in its tracks. These sorts of fossils tell us a lot about the behaviors of ancient animals and where they lived. The final type of fossils are chemical remains, called biomarkers. You find biomarkers in rocks. A biomarker is any measurable substance in a rock that indicates that an organism was present at the time that the rock was deposited. All of these different types of fossils tell us about the history of life on Earth. But let's focus on body fossils, since they are the most common and most familiar to you. How do you go from a living, breathing organism to a fossil surrounded by sediment or sedimentary rock? And what things determine whether or not a paleontologist will find, collect, and study a certain fossil? The science of fossil preservation, or fossilization, is called taphonomy. Taphonomy is the study of all of the processes that affect an organism as it becomes a fossil, from the time of its death to the present. In fact, there are many taphonomic pathways that a living organism can take in order to become a fossil. Think of these pathways like sequences of events. Each event alters the organism in some way. All of the events together transform its tissues into fossilized remains. The pathway that an organism takes toward becoming a fossil is important. Depending on the sequence of events, a fossil may be preserved in any number of ways. Its appearance depends on its taphonomic pathway. Indeed, a single organism can, in theory, leave behind multiple fossils. These different fossils may look like they came from different organisms, but they all originated from the same life form. Before you can understand why some fossils appear so different from each other, we need to explore the most fundamental ideas in the science of taphonomy. Fossils occur almost exclusively in sedimentary rocks. The principle of included fragments tells us that if a rock contains a fossil, then the rock must be younger than the organism that produced it. The organism must have come first, then the rock. For these reasons, The first steps in fossilization are almost always death and burial. Burial is essential. An organism can only become a fossil within sediment that becomes sedimentary rock. Sometimes, an organism dies before it is buried in sediment. In this case, the organism may be transported from its habitat, the place where it lived, to its final resting place, where it is buried. In other cases, organisms are buried alive and die from asphyxiation, suffocation, or other injuries caused by the burial process. Fossils of organisms that were buried alive are some of the best resources we have for studying ancient life, because these fossils are preserved in place. They provide snapshots of organisms at the times of their death. So we can simply look at the rocks in order to determine their environmental habitats. We can also look at other fossils preserved in place to understand how those ancient organisms may have interacted with each other in the past. We can study entire communities and ecosystems in this way. Another important process involved in fossilization is decomposition, or decay, the process of rotting. When an organism dies, its metabolic reactions cease, and its cells no longer protect themselves from organisms that would consume them. They are quickly overgrown by living things like bacteria, fungi, plants, and animals, which break the dead organism down into smaller pieces. Bacteria and other microscopic organisms are the main agents that cause decomposition. When they overgrow a dead organism, they release chemicals and enzymes that dissolve the cells of its soft tissues. Then, the bacteria absorb the fragments of the cells and use them as organic molecules or fuel for their metabolic reactions, like respiration. This process is fast. Decomposition starts almost immediately after an organism dies, and it can completely degrade it over days or weeks, depending on its size. Decomposition destroys virtually all soft tissues of organisms before they even have a chance to become fossils. But organisms aren't just soft tissues. Many organisms have hard parts. These hard parts consist of minerals and substances like calcite, aragonite, apatite, and silica, which do not decay. For this reason, the vast majority of fossils are remains of hard parts and skeletons, like bones, teeth, and shells. Of course, minerals are not indestructible. Indeed, chemical reactions can dissolve or recrystallize minerals in hard parts, destroying them in the process. For hard parts and soft parts to become fossils, they usually have to undergo a process called autogenesis, or autogenic mineralization. Autogenesis is the process in which minerals form within sedimentary rock during its deposition and sedimentation prior to compaction, cementation, and lithification. There are many different causes of autogenic mineralization, but in general, it occurs prior to burial compaction and cementation, when minerals form through precipitation of chemical species found in high concentrations within the water surrounding sediment grains. Importantly, during fossilization, these minerals precipitate around and within organisms. The minerals precipitate inside voids and cavities within hard parts and soft tissues, like the pore spaces inside of bone. One specific example of authigenic mineralization is permineralization. Permineralization is the process of fossilization that occurs when minerals fill spaces within organisms, creating three-dimensional casts of their bodies. Petrified wood, for example, forms through this process of permineralization. Petrified wood is usually composed of silica, the same material that makes up the mineral quartz. After the plant died and it was buried in sediment, the spaces inside of the wood were filled with water, which contained high concentrations of dissolved silica. For whatever reason, a change in the environment caused the dissolved silica to precipitate, and it formed as a solid inside of the wood. In any case, after authigenic mineralization, the fossil and sediment undergo lithification, as the remaining water is forced out of the sediment through compaction, and it is cemented to become a solid sedimentary rock. Along the way, the fossil may be compressed by burial compaction. It may become flattened, depending on what minerals and materials it consists of. At this point, the process of fossilization, for all intents and purposes, is over. That said, authigenic mineralization and lithification do not guarantee that a fossil will survive for someone like you or I to collect. Virtually all sedimentary rocks undergo another process called diagenesis. Diagenesis is the process in which rocks are altered by the high temperatures and pressures that exist deep inside our planet. Lithification is only the first step of diagenesis. After lithification, diagenesis depends on how deep the rocks are buried and what temperatures and pressures they experience deep in the earth. In many cases, diagenesis is destructive. Diagenesis can cause new minerals and materials to form inside of fossils, but it can also dissolve and destroy minerals and materials as well. And it can cause a mineral inside of a fossil to undergo recrystallization. During recrystallization, the composition of a mineral may remain the same, but its atoms are reorganized and rearranged. Recall that the minerals calcite and aragonite both consist of calcium carbonate, but that they have different crystal lattice structures. In many fossils, aragonite undergoes recrystallization during diagenesis. Its atoms become rearranged, and it becomes calcite. For this reason, many fossils of seashells consist of calcite, even though the organism's original shells consisted of aragonite. If a rock experiences sufficiently high temperatures and pressures during diagenesis, it will undergo the process of metamorphism and become a metamorphic rock. Metamorphism is like cooking a sedimentary rock. Most of its features are lost in the process, including most of its fossils. It is very unusual to find fossils in metamorphic rocks, because metamorphism destroys fossils. Beyond metamorphism, there are several other processes that affect the likelihood that a fossil will survive all the way to the present. When sedimentary rocks and fossils are exposed on the surface of our planet, they are slowly destroyed by weathering and erosion. Throughout Earth history, these processes have destroyed most fossils before paleontologists could ever find them, and they continue to affect fossils on the surface of our world today. Clearly, most taphonomic pathways end in the destruction of organisms and fossils. Most organisms do not become fossils, and most fossils are destroyed by decomposition, recrystallization, dissolution, diagenesis, metamorphism, and weathering and erosion. When you really think about it, we're lucky to find any fossils whatsoever. Most individual organisms do not become fossils after they die. And if you or I passed on from this world, it would be very unlikely that either of us would become fossils. It is also probably the case that we do not have fossils of most extinct species. Think about it. Many organisms on Earth today don't even have hard parts like bones, teeth, or shells. So their fossils are extraordinarily rare to begin with. Then, when you consider all the processes that destroy fossils and prevent us from collecting them, it becomes increasingly clear that we will not find fossils for every species that ever lived on Earth. For this reason, you will often hear people say that the fossil record is incomplete. This statement is true. The fossil record is not perfect. Many species are missing. It primarily consists of hard parts, and soft-bodied organisms are missing. And many fossils get destroyed. But there is hope. All around the world, in rocks of various ages, there are special layers that contain extraordinary fossils. We call these layers lagerstätten. One layer is called a lagerstätte. Multiple layers are referred to as lagerstätten. It's a German word. The translation means storage place. Lagerstätten are natural storage places for extraordinary fossils. They come in two flavors. Concentrat Lagerstätten are concentration storage places. They contain high concentrations of fossils. Most of these fossils are disarticulated and broken hard parts, like bones or shells. But the fossils are extraordinary because they occur in great abundance. It is easy to find many species in Concentrat Lagerstätten because the fossils are so abundant. The textbook example of a Concentrat Lagerstätte is a bone bed. A bone bed is a bed of bones that formed from vertebrate animals that kept dying, accumulating, and decomposing in one place. So their bones amassed over time. The Gray Fossil Site, located in Tennessee, generally fits this model. It has produced fossils of fish, amphibians, frogs, reptiles, bears, weasels, badgers, snakes, turtles, rhinos, sloths, rabbits, rodents, bears, beavers, horses, saber-toothed tigers, red pandas, and tapirs. All of these fossils were probably preserved in a pond that formed within a sinkhole during the Pliocene between 2.5 and 5 million years ago. The animals probably came to feed and water in the pond, but in many cases they fell into the sinkhole and couldn't escape before they died. So the skeletons accumulated over time. The other flavor of Lagerstätten are Conservat Lagerstätten, or conservation storage places. These layers are known for their exceptional preservation of body fossils. The layers contain complete, articulated skeletons preserved with remarkable details. They also sometimes contain remains of soft tissues, like feathers, skin, insect wings, insect eyes, jellies, and even the brains and nerves of some animals. Fossils of these tissues don't occur in other layers, so Conservat Lagerstätten provides some of the most complete snapshots of ancient organisms in the entire fossil record. We can study the individual fossils to learn about their soft tissue anatomy and morphology. Or we can examine an entire layer to better understand the ancient community and ecosystem as a whole. Incredibly, many Conservat Lagerstätten date back to some of the most important events in Earth history. The rise of animals and the Ediacarobiota in the Ediacaran period. The diversification of animal life during the Cambrian explosion. The evolution of life on land for the first time in Earth history during the early Paleozoic. And the origin of feathered dinosaurs and birds in the Mesozoic. Thank goodness for Lagerstätten. Lagerstätten help us to overcome the incompleteness of the fossil record. Yes, the fossil record is imperfect. There are many biases. Many species are missing. It primarily consists of hard parts, and most fossils get destroyed before they are ever collected by paleontologists or amateurs. Given these facts, the only thing we can do is remember how fossils form and work with what we got. After all, we aren't getting another fossil record anytime soon.